Everything is working. Is that going to work? Yes! So I've renamed, I read I'm not going to talk about it. I got very interested in how light works, and I discovered that it's not easy to understand how light works. You need to be a theoretical quantum physicist to understand how light works, and I'm not. So I thought I'd instead call this talk, What Does It Take to Be a Scientist? And this book I was reading about light, he says it's unquenchable curiosity and resolute determination. I think that's probably a considerable part of it. And when people ask me, you know, what's the secret of success, as people do, I tell them to keep their eyes on the horizon, their feet on the ground, and their nose to the grindstone. And I think that's pretty good advice, actually. You have to be open. So, actually, what I'm going to do is tell the story of my life, and it's sort of it's a scientific talk, but it's a very old scientific talk because I'm a very old scientist. I was born, as you learned from Janos, in 1943, which means that I'm 82 years old. And I don't have a lab anymore. I don't go to the lab anymore. I don't really have any scientific projects anymore. My job is to look after my wife and do the cooking and wash the dishes and sweep the floor. Oh, and look after the dog. We have a new young puppy who needs a lot of looking after. I should have put in a picture of the puppy. So I grew up in Oxford. My father's office was actually that thing there, and here he is. And he was a kind of medieval historian and librarian. So his friends were all medieval historians and people, experts on manuscripts. And that was sort of good in a way, because we spent an awful lot of time learning Latin when I was a youngster. And that taught me that I was not very good at Latin. So I could give that up. And then one of the things that happened, this is me, would you believe? I think I'm pretty cute, don't you? That's Nicky Bullock, whose father wrote the first biography of Hitler, and Chrissy DeWitt, who came from South Africa. And I never found out who this beautiful young woman who was being Titania. But anyway, I would think I was 7 years old at the time, or 7 or 8, or something like that, and it was in the garden of St. John's College, Oxford, and learning to perform on stage is very useful for being in big events like this. I would strongly recommend it. I mean, all of those of you who are performers of one kind or another will understand that. So I was lucky. The Latin, as I say, I got worse and worse at as time went on, but the science was great, and Gert Sommerhoff was a great teacher. Only one science lesson a week, but he explained things clearly and simply. He told me that understanding science, when you really understood it, was simple, and above all, that it was fun. And those were wonderful lessons. And he also taught me inadvertently that I was a good biologist and a hopeless physicist. So my life course was actually set from a very early age, about 11 years old, I would have said. I knew if I wasn't a biologist, I had no idea what I would be. And so as a teenager, I sort of had my heart set on this, and I hoped that I was good enough. And of course, you worry about whether you have enough determination and whether you've got what it takes, because you don't really know what it takes. You don't really know what it's like until you actually try it. But I had heroes and heroines, and my first real hero, or heroine rather, was Marie Curie, whose biography I read. And if you haven't ever read the biography of Marie Curie, I can't recommend it too highly. I mean, she was an unbelievably wonderful scientist and a fantastic example to us all. And then the next thing, I discovered was the story of penicillin, which I found very romantic, how Alexander Fleming discovered these a mold, a fungus, which could kill bacteria. This is a resistant strain of bacteria along the bottom, but here these guys, they're all dead. They can't get near the mold. But Fleming was a useless chemist, wonderful microbiologist, probably a very good doctor, but a useless chemist. So he never found out what his penicillin was. And that was left to an Oxford team, of whom really the most important people were Chain and Heatley. Heatley it was, in fact, who discovered how to extract the penicillin, how to grow the penicillin, and how to assay the penicillin. Without Heatley, there would be no penicillin, but he was the junior member of the team, and these were the three who shared the Nobel Prize. But it's, again, a very, very inspiring story of how an idea turns into an amazing, life-saving invention. Number one antibiotic. And at school, another strong influence on me was I grew up in Oxford, you see, and there were lectures given by members of the university. I think in the Lent term, January and February, 8 lectures. And the first one were on evolution, celebrating the anniversary of the publication of The Origin of Species. And so that was good. Then the next one was all about the biological effects of ionizing radiation, which were not very well understood at the time, but highly topical in the middle of the Cold War. And finally, somebody from the biochemistry department talked about biochemical pathways, and that really sort of turned me on, because I was quite good at chemistry at school, and I thought this would probably be the thing to do. So I sort of hoped to be a biochemist, but they were terrific. And so I left Oxford, because partly to get away from home, and partly because Cambridge is a place to do science. I mean, one of the things that people don't understand is that 2/3 of the people in Cambridge are scientists, whereas 2/3 of the people in Oxford are not. That's not to say there aren't some bloody good scientists, especially medical scientists in Oxford. I don't mean to put them down by any manner of means. And a great friend of mine was David Weatherall, who was a wonderful physician and a wonderful scientist, and has an institute named after him. Anyway, this was my college. You've already seen a picture of it. We were right next door to King's. I had a friend in King's, and later on in life, I actually lived in a room somewhere about there. So why was Cambridge so good? Well, this is a quotation from Alexander Pope. Nature and nature's laws lay hid in night. God said, "Let Newton be," and all was light. So the tradition was set early on, but after that, James Clerk Maxwell became the first professor of physics. Thomson discovered the electron, and then, as we heard earlier, Rutherford and his team discovered the atomic nucleus, the proton and neutron, and first split the atom. I mean, I used to walk through. This all happened within an area not much bigger than this enormous arena, actually. It was absolutely extraordinary. I used to walk past it. And if you were sort of feeling good about yourself, you'd say, well, I'm in this great tradition too. But more often than not, things weren't going so well, and this was actually quite a burden to carry. It's sort of something you couldn't live up to. And then again, it was in the physics department that Crick was really a physicist. Watson was the bird watcher. And they worked out. Well, actually, this is a little bit unfair. Watson and Crick did not discover the structure of DNA. It was actually Brown and Todd who worked out the structure of DNA. They were chemists, but they did it just before. I mean, Watson and Crick needed to know the chemical structure of DNA, and this used to annoy Todd. He said, I was the one who discovered the structure of DNA. Watson and Crick merely determined the configuration of the two strands. And so And then Kendra and Pruitt worked out protein structure. So again, you know, this was a wonderful place to be. And I joined Cambridge University in 1961. 1962, they won the Nobel Prize. So it was in the air, and we had lectures from people like Sidney Brenner and Fred Sanger and co. I mean, it was, you know, and messenger RNA was discovered when I was an undergraduate. And I remember my friend Bridget coming in, brandishing a copy of Nature, saying, Tim, they've discovered messenger RNA. You know, this was a good time to... And later on, I got to know both of these characters. So, and here is Kendra and heme. So now I want to start on a sort of scientific. So here's hemoglobin, which is really 4 of these chains put together, 2 alpha, 2 beta, and 4 hemes. And my career really started, my original scientific project was a complete failure, which was good. I sort of learned where the pipettes were kept in the lab. But I went to a scientific meeting, which was very close by. It was in the chemistry department. And these 2 talks, one by Borsuk, the other by Ingram, planted seeds in me. And it was actually Ingram's talk that really set me going on the path. So Ingram was concerned with the question of, well, no, I should backtrack. Borsuk had discovered that if you starved young red blood cells of iron so they couldn't make the heme, not only did the heme synthesis stop, but also the protein synthesis. And this they found very surprising. Ingram was sort of following up on that and trying to find out how, and he showed data that suggested that the way it worked was that the ribosomes got onto the messenger RNA, and then they went until the point where the heme should come in. And at that point, they would stop and wait for the heme. If the heme wasn't there, they couldn't move on. And then when the heme came on, they could finish making the chain. And I thought, gosh, that's interesting. Went back to the lab and explained to my friends how it worked. But my friends, being cleverer than me, realized that actually he'd completely misinterpreted his data. So in a step that I cannot now reproduce, I don't remember what happened, we decided to find out for ourselves. Did ribosomes form Qs or not? And when I say we, there were 2 important people here. Lou Reichart, who is a distinguished neurobiologist these days, or probably like me, retired, and Tony Hunter later joined. Lou knew how to make reticulocytes. These cells here are still making hemoglobin. And if you feed them radioactive amino acids, they will make radioactive globin. And in fact, Lou was trying to get heme to control protein synthesis and didn't succeed at that time, again, for reasons that I can't now reproduce. So Tony and I collaborated, and we showed that actually the ribosomes didn't form Qs, and we found out 1 or 2 other things, which I won't go into, and it was quite successful. But along the way, I went to my second scientific meeting where I met this man here, Irving London, who was interested in exactly the same thing. How did the heme control globin synthesis? And I ended up going to his lab as a postdoc. I actually spent a sabbatical 3 months as a graduate student in New York, and I decided that I liked living in New York. I liked his lab, I liked the people there, and America at that time was barely involved in Vietnam as far as we knew, and it was a good place to go. And he offered me a place to work, and so I thought that was good. And by the time I got there, we'd worked out how to make cell-free preparations which could actually make proteins at pretty much the same rate as the intact cells. And if you left out heme, protein synthesis curled over and died quite early on, and then went at a low rate, typically 10% or something of the proper rate. And moreover, if you add back the heme after the thing has curled over and died, it kind of recovers. It doesn't recover all that well usually, but you can see in this experiment it covered about 50%. So a reversible control. So how did it work? Well, this was a very reproducible result. But we didn't really understand enough about how protein synthesis worked to be able to figure out what was going on. And so I got a bit discouraged and started doing naughty projects after hours of my own devising. And one of the projects was to look at the translation of poliovirus RNA, possible at Einstein because there were people who worked on polio. So I got some polio RNA, added it to the reticulocyte lysate, and it didn't make anything. So I reasoned that the reason it didn't make anything was because polio doesn't normally infect red cells, and if, however, since it did infect HeLa cells, HeLa cell extract must contain the things that it needed to translate it. And so I added some poliovirus-infected HeLa cell extract into my reticulocyte lysates and got a very surprising result, which was that, again, nothing happened for the first 10 minutes or so, and then suddenly protein synthesis shut off again. And we wondered what the hell this was. Now, by this time, Nixon was bombing Cambodia secretly, so quite often we used to go down to Washington at the weekend on protest, and I remember being tear-gassed outside the White House, which was rather unpleasant. We had to seek refuge in a nearby Hilton hotel. It was awful. And it was awful also because one felt so powerless that here was the American government doing something of which you so strongly disapproved, and here we were doing experiments in the lab. It didn't feel right in a funny kind of way. I mean, I don't know what people feel now. It's sort of interesting, and we may be going back to that kind of era, actually. Anyway, Ellie and I collaborated, and we found out it took us a while to figure this out, because what was it that was causing this shut-off, and what was it? You couldn't digest it with any enzyme that we found. It was resistant to ribonuclease, it was resistant to DNase, it didn't do it with protease. The only thing that did destroy it was strong acids and bases. It was also very big. So we were puzzled until we realized that double-stranded RNA, which looks very much like DNA, but you can see it's not the same, was resistant to ribonuclease. And so I asked Jerry Hurwitz, who worked upstairs, what could I digest this double-stranded RNA with? And he said, oh, you use micrococcal nuclease and don't forget to add the calcium. And that was very helpful later on. And indeed, when you added micrococcal nuclease and calcium, that destroyed the inhibitor, and so it was double-stranded RNA. And by this time, we knew that actually what was happening was that an inhibitor formed which blocked initiation of protein synthesis. No block down here. The block was right at the beginning. And when we calculated how much double-stranded RNA you needed to get this inhibition, one molecule of double-stranded RNA could inhibit synthesis by about 20 million ribosomes. So it had to be catalytic. But we didn't, we were sort of stupid and slow and didn't really put two and two together. So I guess I had one Nature paper and a Journal of Molecular Biology, but it wasn't a very successful postdoc. I don't think I could have got a job after that. So I slunk back to Cambridge, where I still had a college fellowship, and I joined forces with Richard Jackson. Jim Watson writes in one of his books, always try to work with people who are cleverer than you are, and this is very good advice for somebody like me, and we made a very successful partnership. Alas, he's no longer with us, and but we began to figure it out. And the first thing we figured it out, Richard and Tony had discovered that protein synthesis used a special transfer RNA to start with, called initiated tRNA, that carried a methionine residue. And we switched to our label from, I think, leucine to methionine, which turned out to be a wise choice. And when that was done, we discovered that there was a complex between methionine tRNA and the small ribosome subunit, which disappeared when the heme wasn't there. And it disappeared before the inhibition set in. So you could deduce that whatever this thing was, it was a precursor to protein synthesis. And we had tremendous difficulty publishing that, because everybody claimed. We were claiming that what happened was that the small subunit of the ribosomes bound the initiator tRNA, and then that went looking for the start of the messenger RNA. And then when it found the start, it stopped, the big subunit would come on, and everything would get going. But people said that they were so blinded by prejudice that tRNA only bound under the instruction of messenger RNA, that putting it round the other way challenged their most cherished assumptions. And so we were given a very difficult ride in publishing this, but we succeeded in the end, and of course we were right. And then the lab burned down, and that was the best thing that could ever have happened in retrospect. At the time it was rather shocking, and it took about 6 weeks to restore the lab to normality. And as luck would have it, our new lab, our temporary operation, was in the Addenbrooke's Hospital in an unused hematology teaching lab across the road from the famous LMB, where there are already 21 Nobel Prizes in biology. And Max Perutz, who was the director of the LMB, was very kind. He said he could use his stores and use his canteen. And that was a fantastic gift, because in the canteen were all these wonderful scientists. And they weren't so high and mighty. You know, they would sit down at lunch and explain what they were doing and thinking about, and you could collaborate with them or their graduate students, and it was fantastic. And as a result of that, I think, we had been very confused, actually. I mean, I won't go into what all these things are, but basically, you know, we were studying all kinds of funny things that we couldn't understand and couldn't make sense of it. And out of this fog came the answer, a very simple answer, which was that what happened if you left out the heme or you added double-stranded RNA, a protein kinase turned on that phosphorylated this protein here, which had just been isolated by Theo Stählin in Basel, and was the initiation factor that carried the initiator tRNA. And so everything suddenly went into place. And this was my first real taste of scientific blood. You know, we'd been working on this problem for almost 10 years, and it turned out to have a very simple answer once we got it. The terminal phosphate of ATP reacts with a protein catalyzed by an enzyme to make protein phosphate and ADP. And here's an example of a protein getting phosphorylated, and this form of the protein without the phosphate is completely dead. Stick that phosphate on, you see that the changes in configuration around here are really very modest, but it makes an almost infinite change in the activity of the enzyme. So that was all well and good, and the problem was now what to do next, because one of the problems, I think, if you're a scientist, if you solve a problem, you don't have a problem anymore. And in my experience, actually finding a scientific problem is difficult, because you can all think of problems to solve, but, you know, could you actually solve them? And of course, there are trivial problems that you can easily solve. And one of the things I remember was another part of Borsuk's talk about how when sea urchin eggs were fertilized, they turned on protein synthesis. And I thought it would be fun to work on that. And I invited Tom Humphreys to a meeting we organized in Cambridge to tell us what was known about that in the intervening 10 years since Borsuk had talked about it. And the answer was not very much. And it turned out that Tom was a very keen cyclist. And he asked if he could rent a bike in Cambridge, and I said, no, but you can borrow mine, and told him where to go. So we became friends because of our shared enthusiasm for cycling. And at the end of the meeting, Tom said, why didn't I come next year and help him teach the embryology course in Woods Hole, which I already knew as being a very beautiful place, because my previous boss, Irving London, had a house there that he used to go to in the summertime. And it's a sort of mecca, summer mecca for biologists, and the labs are over here. So I jumped at the chance and learned about sea urchin eggs, and here I am getting eggs out of a sea urchin with a 12-volt alternating current from a doorbell transformer. And if you do the same thing to a male sea urchin, you get this sperm. The sperm is very good stuff, actually. You can put it in the fridge and it keeps for ages. And you just dilute it down, add it to the eggs, and they get fertilized. And just as Borsuk said, unfertilized eggs essentially make no protein at all. We fertilize them, there's a little lag, and then it takes off, and there's a good rate. And I wanted to understand what this switch mechanism was. And I made absolutely zero progress the first year. And in the second year, oh yes, and the other thing which I learned in Woods Hole was if you look down the microscope, if I add some sperm to these eggs, you'll see what happens. This is very speeded up, but you get these amazing, beautiful, synchronous. All you have to do is just mix the eggs with the sperm, and they start dividing, as well as increasing protein synthesis. So that was good. And then that second summer I went back, I collaborated with Joan and Eric on clam eggs. And clam eggs didn't increase the rate of protein synthesis. They did something much more interesting. They started making new proteins. You can see before fertilization, these proteins here, A, B, and C, well, maybe there's a little bit of C, but not much. But A and B are really absent. And that's sort of what they look like. So that was good. And that summer, I heard a crucial seminar by John Gerhardt, an excellent enzymologist who really discovered allostery. And he described experiments that he was doing in his lab on frog oocyte maturation. And if I add some progesterone to these oocytes, you'll see what happens, I hope. Yeah, there we go. It's not very dramatic. But he described the things that had been done a few years before by Mazui, which was that progesterone turns the oocyte, that's the cell that's going to be an egg, into a fertilizable egg that comes out of the cloaca of the frog. And it's actually arrested in second meiotic metaphase. And what Mazui discovered was if you suck a little bit of cytoplasm out of this and inject it into new oocyte, then it very rapidly makes this transition. And whereas this transition requires new protein synthesis, this one doesn't need new protein synthesis. So I thought, gosh, what a delicious problem. What is MPF? And the trouble was that John, who was a very good biochemist, had been unable to make much progress with this because it was so unstable. But it was destroyed by heating and destroyed by protein digestive enzymes, so it probably was an enzyme. And the idea that there could be an enzyme that catalyzed cell division struck me with amazement, because cell division is so complicated. How could there be an enzyme that catalyzed that? No idea. You just couldn't conceive of what it could be. And then more experiments showed that MPF was not just in frogs, but first starfish, and then finally human and yeast, all have MPF that work in frogs. So MPF is not just a sort of curiosity of frog oocytes. It seems to be a universal factor, which presumably does the same thing in all organisms. I think these are before the days of DNA sequencing, we didn't really fully appreciate the commonality of all nature. You know, the idea of how closely related we all are. And then it took I heard that seminar in '79. It wasn't until '82 that I did this experiment. And this is the Nobel Prize winning experiment, which I correctly interpreted and also ran into John Gerhardt the very evening that I developed this gel, and he told me a fact which was absolutely electrifying. And I don't know if you can see what attracted my attention. It's a very simple experiment. You fertilize some sea urchin eggs. When you're sure they're all fertilized, you add some radioactive label, radioactive methionine, and then you analyze the results on a one-dimensional SDS gel. You dry it down, you put an autoradiograph. So these black bands are radioactive protein. And I think you can see here, this protein here is the most abundant early on. And it gets stronger and stronger. Other proteins get stronger and stronger, but they're much less strongly synthesized. And then it sort of faded away. Gosh. Proteins can't fade away like that. And the way the experiment was done meant it was, I mean, this is an incredibly well-controlled experiment. There's only one possible interpretation. This protein inside the cell, and it alone seems to be going when all the other ones are coming. So at the time, that was inconceivable. So inconceivable that nobody had ever suggested theoretically that it might happen. And the idea that the cell division cycle needed something to go away, to be destroyed by proteolysis, had not even been suggested as a theoretical possibility, because it would, at the time, have been conceived to be totally impossible. But there it was, in black and white, so to speak, before my very eyes. So what do you do? Well, the first thing was we repeated the experiment, being careful also to measure the first division, the second division. And you can see here is our protein coming up, again, very strong, disappears, comes back again, disappears again. And it kept on doing that as long as the cells divided. And moreover, if you stopped them dividing, say by inhibiting DNA synthesis, it just came and never went away. So, gosh, what is it? And then we looked in clams, and it turned out that these proteins we called A and B both did this, and we hadn't figured that out until we'd seen it in sea urchins, because we never did that very simple experiment, just taking it. You can't see something go away unless you take a time course. That's I think, the lesson here. Now, so I was super excited by this, and I knew immediately that I was hot on the trail of MPF. But I also knew that I was totally ignorant, knew absolutely nothing about the cell cycle. Now, very luckily for me, nobody else knew anything about the control of the cell cycle in 1982. So I talked to people and to see if they could see anything wrong with my suggestion of how it was going on, and nobody could pull any holes in it. And I knew I was right, I mean, that there was this protein, but figuring out what it was and how it worked was very difficult. So we had to do a lot of studying, and here is the cell cycle with mitosis and DNA replication and gaps in between. And of course, DNA is really the key to all this. Sidney Brenner said, a moment of absolute enlightenment, the reduction of biology to one dimension. Absolutely great. And he'd been reading von Neumann, a Hungarian, on cellular automata. How could you design a machine that would self-replicate? And von Neumann said that the machine had to contain a tape with instructions how to make it, and the machine would have to copy the tape each cycle, and would have to have a mechanism for not only for building the machine under the instructions of the tape, but also for distributing the tape to each member. And so, G1 phase is preparing for copying, then you copy it, then you prepare for insertion, and you put the two identical copies of the tape into the two daughters. And because DNA has two strands, cells divide in two. Very simple. And what was known mostly came from yeast genetics, and Lee Hartwell and he focused particularly on CDC28, because that seemed to control three different things, so it's probably a regulator rather than, say, an enzyme that catalyzed DNA polymerase or something like that. And Paul Nurse read his papers and studied a different kind of yeast, and Paul focused on a gene called CDC2, because Paul found that if you knocked out CDC2, the cells couldn't divide. They just got longer and longer and longer until they ran out of energy and died. But there was also an allele of CDC2 that actually accelerated cell division and made smaller than usual cells. And then finally, CDC2 and CDC28 seemed to control different cell cycle transitions, and it came as a surprise when DNA sequencing revealed that they were actually the same thing. That caused a considerable puzzle. And Paul found you could either kill it or make it hyperactive. Its sequence suggested that it controlled the protein kinase, but if you tested it, it didn't have any protein kinase activity. So Paul said, it's not a protein kinase, Tim, we've tested that. No, it must be some enzyme that uses ATP to do something different. And from this, there was no suggestion that cyclin fit in anywhere or that proteolysis was contained, because yeast genetics can't tell you that kind of thing. It can tell you what the components are and where they fit together sort of in a logical scheme of things, but they can't actually tell you anything about the physiology. So I had two wonderful graduate students, John Pines and Jeremy Minsal. Here they are flanking nurse, clutching a glass of wine. John and Jeremy succeeded in cloning cyclins, first from sea urchins and then from frogs. We were very excited when we found cyclins in frogs too, because then we knew they would be in humans. And the next development really came from a completely different quarter. Fred Locher, who'd worked with Mazui, actually, and Jim Mahler, worked out that if you made a cell-free preparation, this is pure cytoplasm from frogs' eggs, which will synthesize proteins, and if you add nuclei to them, will assemble beautiful meiotic spindles with poles, green poles and purple chromosomes all lined up. And were you to add a bit of calcium to this, they would actually do metaphase. And using this as their assay, they managed to purify for the first time MPF and found that it had two subunits, a larger one, the molecular weight of cyclin, and a smaller one, the molecular weight of CDC2. So and that if you added gamma P32 ATP, it was a protein kinase that would phosphorylate histone H1 and also the cyclin itself. And when I saw this, I said to Jim, as soon as we have antibodies, we'll send you them and we'll prove that this subunit is cyclin, and they did the same for CDC2, and so it was really easy. So you needed cyclin to turn on CDC2, and the key came in this loop of protein here. In the unactivated, uncyclin complex form, that loop is sort of over the active site of the CDC2, so it can't work. And the role of the cyclin is to pull that loop away and activate the enzyme. You also need to phosphorylate that loop, as I showed you earlier. So what had seemed totally mysterious became very simple and easy to understand. The cyclin accumulates, turns on CDC2, that catalyzes entry into mitosis. The cyclin is destroyed, that turns off the CDC2, the phosphates are all removed, and the cell exit mitosis. And, you know, I mean, what took us so long? And it's a very interesting question of what took us so long, because some very clever people, much cleverer than me, worked on this. And we were very slow, actually, because we were very prejudiced about protein kinases. Up to that time, the only protein kinase was known had a companion subunit, which was an inhibitor, and turning it on meant inhibiting the inhibitor. Here we had an activator. And, you know, I mean, yeah, it's still... Anyway, when luckily, hardly anybody worked on the cell cycle in those days, so we had complete freedom for about 4 or 5 years, I suppose, to do things properly and work things out. Then the minute people could see how it worked, they all piled in, and there was an awful lot of fluff and confusion in the literature, but it was super fun, I can tell you. So here it is. You can almost see the cyclin go away. I do have a movie that's less dramatic, but they're so beautiful, these things. I mean, it's just, yoing, doing. Isn't that lovely? You know, and it turns out the cyclin gets destroyed about 90 seconds before those chromosomes come apart. And, you know, if you inhibit its going away, then everything goes horribly wrong, and they stay in mitosis forever. So this was immensely satisfying, and it's not the end of the story, but I don't have time to tell you any more, so we'll stop there. And I think the moral of the story is that, you know, the road may seem fairly obvious for the first bit of the way, but you have no idea where it goes over the hill. And what you have to do is simply follow your nose and go where the experiments carry you. Thanks very much for listening.
Thank you, Tim. And since we are a little bit delayed, we have time for 2 questions, please.
if there are any. There's one there. I can see a hand go up.
Thank you.
So my question
Just
would
please
be,
stand up.
Stand up. Stand up.
my question would be that, what do you think of manipulating the cell cycle in humans, for example, in anti-aging research, and what do you think of this?
Well, the trouble is if you inhibit this, well, one of the things I learned in that lectures on ionizing radiation when I was 18 years old was that if you stop cell cycles, you die horribly in a week, yes? So it's not a good idea to inhibit the cell cycle because your guts drop out, you suffer from terrible anemia, and it's a, if you want, yes, I wouldn't recommend to read the book by the Japanese physician who did write a book about what he saw in Hiroshima. I mean, it's just awful, just awful. And people say, well, now you could cure cancer because all you could do is to, you know, inhibit the cancer cell division. But how do you do that without inhibiting all cell division? The answer is nobody's yet figured out a way. And the one thing that's never wrong with cancer cells, they do know how to divide very well. You know, that's not their problem. Actually, the main problem with cancer that we still don't really understand, it's not a matter of cell division, it's about cell growth and division. And the control of cell growth is very poorly understood. I mean, a lot is known, but it's not really terribly well understood. And I always like to say that the great unsolved problem in biology is how you stay the same. And I always like to think of your nose. Because if you think about your nose, you have an absolutely new nose. There's not a single molecule in my nose that was there 10 years ago, right? All been replaced, except maybe a few collagens, but that's by the by. Yet my nose is pretty much the same shape as it was 10 years ago. So why doesn't it, what keeps it the same length? You know, why doesn't it shrink away, or why doesn't it start growing? It doesn't. It knows how to be a nose. And when if it got cancer, I mean, nose cancer I think is extremely rare. But if it did, you'd immediately recognize, because you'd see it would be a horrible sort of suppurating thing, which didn't look like a nose anymore. So it sort of forgets it was a nose. But I don't know, you know, how. I mean, these are very common things, like your skin is dropping off all the time, you know, or red cells. You know, you make, I think it's 2 million new red cells every second. And if you stop that, it's very bad. But, you know, making exactly the right number of red cells is really important, because too many and your blood won't flow in your veins anymore. Too little and your brain won't get enough oxygen anymore. And all those homeostatic mechanisms are, I mean, in some cases very well understood, but I think not in all. I don't think they're very well understood in the nose, for example. That wouldn't be a very sexy sort of thing to work on. I don't know whether that remotely answers your question.
I think it doesn't. Thank you so much.
Okay, good. Anybody else? Here, right at the front.
Thank you for the wonderful talk. Is science just? So Heatley and all those not forgotten heroes, but who didn't get the same recognition as the others?
Well, I think there are some sort of winners and losers, I suppose. I mean, yes. But, you know, Nobel Prizes aren't everything. And everybody knew what Heatley did. I met Heatley right at the end of his life. I had to open a lab in his name in Oxford. And that was really quite funny, because he'd worked with Joseph Needham in Cambridge. And he was wheelchair. He was about 92 years old and rather demented. I said, "So you worked with Joseph Needham, did you?" "I may have," he said. It was kind of sad. But if you read this story, there's a wonderful book called The Mold in Dr. Flory's Coat, which I would strongly recommend for the history of the development of penicillin. It's extremely interesting. And Heatley gets full recognition there. I mean, the funny thing about that story was that actually at the time, it was Fleming who was the apparent hero, and For various reasons, not entirely clear, the Oxford team kept a very, very low profile and took very little credit for what they actually did. But the Oxford team were really the ones who were the real heroes. I mean, they did all sorts of extraordinary things that I don't have time to go into. You better go on, I think, huh?
Just please come back to the stage.
Ah. Thank you very much. There we go. Gosh, look at this. I have a room full of these medals. If you win a Nobel Prize, you get a lot of medals, and you also get a lot of ties, it turns out. I have a drawer full of ties as well. But I don't wear a tie. But that's okay. Thank you very much.